We go. Hi, I'd like to welcome you to this webinar entitled The Use of Lipid Tracers to Track Fatty Acid Flux in Humans and Animal Models in NAFLD. My name is Elizabeth Parks, and I'm at the University of Missouri School of Medicine, Columbia, Missouri. Joining me online today are four isotope experts. We have Henry Brunegraber from Case Western Reserve University. Steve Previs will be joining us from Merck & Company. Joanne Kelleher, who's affiliated with MIT, and Owen McGinnis from Vanderbilt University School of Medicine. This is the first talk of a three-part webinar. Next month, Dr. McGinnis will be speaking on tracing hepatic glucose and glycogen metabolism in metabolic disease. And then in November, Matt Merritt from the University of Florida College of Medicine will give a talk entitled Central Metabolism is a Target of Pioglitazone Action. All the faculty here and others are all part of an annual isotope tracer course funded by the NIH. This year, the course will be offered in April, and the lower right-hand corner has the link where you can get more information. I'd like to start with my disclosures. All of the methods I'm going to present today were developed in my laboratory, and none of the topics I'm going to discuss are the subject of any of this consulting. So what are we going to do? We're going to talk about the contribution of different fatty acid sources to liver lipid in humans and animals. We're going to discuss the role of lipogenesis in lipid accrual in the liver and in non-alcoholic fatty liver disease. I'm going to trace adipose fatty acids and dietary fatty acids into the liver. So this is a schematic with the liver at the center. And there are three sources of fatty acids we're going to talk about today that can serve as sources for triglyceride secretion. The first one is the plasma-free fatty acid pool. And in the fasting state, your adipose is releasing fatty acids for energy. About a third of those fatty acids go to other tissues in the body. And for someone who's resting quietly and fasting, that's a key source in the fasting state. In the liver, 2/3 of those end up in the liver, and they can be either esterified to triglyceride and stored, burned in the mitochondria, which is not shown here, or they can be immediately repackaged into a lipoprotein particle called VLDL and resecreted back out into the plasma. A second source when we eat dietary fat is that these lipids are packaged into chylomicrons in the intestine and sent out into the body. The chylomicron drops its fatty acids off into other tissues. It becomes smaller. We call it a chylomicron remnant, and then it's taken up by the remnant receptor in the liver. Once there, those fatty acids can have the same sorts, fates, storage, mitochondrial oxidation, or recycling through VLDL. The last source we're going to discuss today is the process of de novo lipogenesis, which are new fatty acids made in the liver from dietary sugars. When we eat carbohydrate, those sugars come in through the portal vein into the liver. This process is slower than the other processes of recycling of fatty acids from the diet or the adipose tissue, and they enter a stored pool at first. So let's look at de novo lipogenesis across species. On the top here are various species, and on the bottom row are the sources of carbons that serve to make fatty acids in these species. Primates eat, actually in the wild, very low-fat diets. So primarily their energy is coming from carbohydrate, and they consume grains and fruits. Most of their lipogenesis is coming then from glucose and fructose carbons, and de novo lipogenesis occurs in the liver and the adipose. The same is the case for rodents. As most of you know, chow is made up of grains, and these starches provide glucose as the source for DNL. The same is the case for rabbits. Pigs are omnivorous, just very much like humans. Our lipogenesis is much lower. The sources are glucose and fructose. And in pigs, most of de novo lipogenesis is occurring in the adipose tissue. Dogs are a species that start to highlight the fermentation capacity in this biochemical pathway. The grains and the fibers that they consume can be fermented in the colon, and acetate then made from the microbiome provides the source of carbons. Cats are the same, being obligate carnivores. In birds, most lipogenesis happens in the liver, and their diets are coming from starch. And then in cattle, sheep, and goats, we've got fermenters, and acetate provides the source of carbons for fatty acid synthesis. We can label this pathway of de novo lipogenesis with isotopes by feeding them, infusing them, giving them as a bolus, IV or IP as well. For years, radioactive water and carbon-14 glucose have been used to track this pathway. And what I'm going to highlight today are the stable isotope use of deuterated water, carbon-labeled glucose, and carbon-labeled acetate. So when these isotopes are given and they come into the hepatocyte, this also occurs in the adipocyte or the mammary cell that's making lipids, glucose comes in very far away biochemically from fatty acids. So when we label sugars to try and find that label in the fatty acids, those sugars can undergo very many fates. And so labeling the carbohydrate is not the most efficient way to label de novo lipogenesis. Glucose can end up in glycogen. It can be burned in other tissues. In the upper right-hand corner, you can see water coming in, and it exchanges with NADPH hydrogens, which are incorporated into the growing fatty acid chain. When we infuse or feed acetate, to the extent it clears to the liver, it gets a CoA put on it, and then it is incorporated through malonyl CoA. So the fatty acids that are made through de novo lipogenesis then can be packaged into VLDL and secreted into the plasma. And we showed a long time ago by labeling up individuals who are having a liver biopsy, we measured the label in the liver tissue and also in plasma VLDL. And we found that the various sources of fatty acids in the liver, triglyceride, were mirrored in VLDL triglyceride. So these days, many people label animals and humans and look in VLDL triglyceride to determine the packaging that's going on in the liver. When we compare mice to humans, and I get this question all the time, the rates of lipogenesis are faster in the liver compared to the adipose in a mouse, but the overall quantity of fatty acids made in adipose is greater because they have greater adipose mass, just like us. The typical mouse diet is mostly carbohydrate. And so if one calculates, you can look at your chow, figure out how much fat is in chow, and calculate body fat accrual on a growing mouse. And you will find that the amount of fat they're eating every day is not enough to support their body fat accrual as they grow, because most of that body fat is coming from carbohydrate. In healthy humans, liver triglyceride concentration is typically low, and its turnover is really fast. We've calculated that in healthy young men, the liver triglyceride pool turns over in about 4 hours. Compared to the adipose tissue, which your body fat could take about a year to completely be used up and replaced, which is a bit of a depressing fact. The sheer mass of triglyceride in adipose is so great that any label that gets there can get diluted. So it's very difficult to measure adipose lipogenesis in humans. You have to label for almost a month. And then we also, or almost 2 months, I would say. And then in humans, we eat a mixed diet. So typically our lipogenesis is low. On a low fat, high carbohydrate diet, de novo lipogenesis increases in humans. And then my lab has also shown that lipogenesis is much higher in individuals with insulin resistance. And we found that this is due to both excess carbon flux through glycolysis and also the stimulation of insulin in these insulin resistant subjects. So let's look at a mouse timeline to label up liver fat. This experiment took us about 20 days the first time we did it. We acclimated the mice to meal feeding during the dark cycle. And I'm going to take a minute to describe this because with respect to rigor and reproducibility, this has been a key component of our animal experiments. We basically remove the food availability from the mice in the light cycle. Mice eat about 25% of their food at night, 75% 25% of their food in the light, 75% in the dark. And so when you remove the food in the light, they lose about 1 gram of body weight in the first day. Then they figure out, oh, they have to eat all their food in the dark, and they regain that weight, and then they grow normally. The importance of doing this is if you're interested in fed state metabolism and also in fasting metabolism that is not stressed. When we acclimate our mice to eating their food in the dark cycle, and it could be 8 hours of availability, 10 hours, 12 hours of availability in the dark cycle, what happens then is that during the light cycle, they aren't looking for food. And you put the food in the morning and they all begin to eat at the same time, such that if you want to capture fed state metabolism, you can then kill your mice 1 hour or 2 hours into the dark cycle. And you really only need 2 or 3 mice per time point because the variance is so small. The biggest advantage of meal feeding then is also in the light cycle, which is the fasting state. You can kill these animals for an experiment 3 hours to 4 hours in the fasting state, and all the animals will be exactly the same amount fasted, and they won't be running around the cage looking for food like they are if you've just removed food for the first time. So we've done this experiment where we add our isotopes to chow. In this case, we used 1,13C acetate in the chow, and that measures lipogenesis. And then we've used tripalmitin, which is perduterated. It's very heavy. It's 31 heavy for every fatty acid. We've killed 3 animals per time point at day 0, at the end of 1, 3, 6, as you can see here. And then we've killed 3 animals in the fed state. We've looked at all of the tissues. I'm going to show you data first from inguinal triglyceride palmitate. This was an experiment where we were looking at the tick snip mouse, but you can kind of ignore that. Both the wild type and tick snip were exactly the same here. And you can see that adipose fats made from lipogenesis are being labeled and slowly rising. This is what lipogenesis looks like in the liver. And the asymptote here is about 40% of liver triglyceride palmitate was being made from de novo lipogenesis, with the 2 strains not being different. These are the curves from the dietary label, a beautiful curve coming up to steady state. And from this, we determined that we could have done this experiment probably for about 10 days or so to get complete turnover of liver triglyceride. If you look at the y-axis here, you can see that in this experiment, only 4% of liver triglyceride palmitate was coming from dietary fat on chow. So we've also done this experiment where we've repeatedly sampled in many, many mice over time. And here are the data. So these are the liver triglyceride pool, and in this case from dietary fat. The pink values are from the fed state, and the closed are from the fasting state. So this on day 1 is the residual label in the liver from the mouse eating labeled dietary fat the day before. So here come all the data. Again, you can see, you can calculate the turnover of liver triglyceride from dietary fat. And in the fasting state, 3.6%, and in the fed state, 4.4%. This turnover was coming from dietary fat. So that was discussing both the lipogenesis and the dietary fats. And now I want to label adipose tissue fatty acids in a mouse. And for this, I'm going to show you an acute infusion study. This is not days, this is over hours. And this is an old study in the ApoB48 knockout mouse. We placed a jugular catheter very much like they do at Vanderbilt all the time. The mice recover, we fasted them, and then we did a 3-hour infusion through the jugular with 13C4 palmitate on albumin. Fractional triglyceride synthesis in palmitate in the genetically modified mouse was 12% in 3 hours. One can then put in the amount of liver weight, the amount of triglyceride in the liver, how much palmitate is there. And when we performed this calculation, we got 19.5 nanomoles of fatty acids turnover per minute in the liver. We repeated this study using tritiated water, and we got exactly the same thing with a bolus shot. We've compared a number of methods for liver triglyceride turnover and found them all to give very similar values. One last point here, under the infusion, we could put glycerol in there to look at adipose glycerol release, or you can put acetate, which, as we did in this experiment, and also measured lipogenesis at the same time. So all of the kinetic methods that we do in humans have been established in multiple animal models. We've done studies in rodents, pigs, and primates. So let's turn to performing this same free fatty acid infusion in humans. Just generally, how do we do that? We're going to mix palmitate, an isotope, with human albumin and infuse it over a period of hours. And how long the infusion should be will depend on what it is you're interested in measuring. It's extremely useful, this technique, to measure three things all at the same time. You can measure adipose release of fatty acids, and this dilutes the label. So from this, you can calculate the rate of adipose fatty acid release. In other words, you're infusing at a steady state a labeled isotope, and we might do this into one vein in one arm. The other arm, we're drawing blood, and the cold or unlabeled palmitate that's in the blood is coming from the adipose tissue. The more your adipose is releasing, the more you're diluting the label. And this is the premise of isotopic studies. At the same time, this palmitate can clear to the liver. And as I've said, it's immediately resynthesized into triglyceride, which is packaged into VLDL, and then that's put in the blood. So we will draw blood and isolate VLDL and look for the triglyceride palmitate label in there. The triglyceride then can be used as that shape of that curve, as I'll show you, to calculate liver triglyceride secretion rates and also the turnover of this lipoprotein particle in blood. Lastly, if you wait long enough and the VLDL particles are being made the entire time, let's say 8 hours, you will clear VLDL and replace it such that all of the particles in blood have been made new since you were performing the experiment. If you go long enough, you can then figure out the percentage of VLDL that's coming from the plasma free fatty acid pool. And I'll show you what that looks like. So on the x-axis is time from starting the infusion of the free fatty acid. And on the y-axis is the enrichment that we observed. So you can see the free fatty acid pool turns over very, very quickly. And the minute we turned on the infusion, in this case, in the right arm of this patient, and we drew blood in the left arm, we could immediately see the label starting to rise and then come to steady state. So that free fatty acid pool has turned over very, very quickly, and we can calculate the release rate of adipose palmitate. At the same time, that label is going to the liver, it's made into triglyceride, and it's resecreted. And so the VLDL particles are being made, they're labeled, they come into the blood, and they're replacing cold, old particles that are there. And so we came up to steady state. So if we calculated these two enrichments, and we put the palmitate in VLDL enrichment over the free fatty acid enrichment, in this case it was 1.75 over 2.60, we can calculate that 67% of VLDL triglyceride palmitate is coming from the plasma free fatty acid pool clearing to the liver. And notice that's not 100%. In lean individuals who have very low liver fat, we get 100%. This individual had stores of fat in the liver and was also probably making fatty acids through lipogenesis, as I'll show you next. Here is two similar curves. On the left-hand side, a control lean subject. On the right-hand side, a hypertriglyceridemic subject with metabolic syndrome. The shape of these curves gives us the VLDL triglyceride production rate, and we can model that. The steady state asymptote, which in this case is the percent enrichment at the top, gives us the source of VLDL triglyceride made in the liver. And in this case, this is coming from the plasma-free fatty acid pool. So what's happening here? Well, this is me being a curious postdoc, and I wanted to know what happened when you turn off the free fatty acid infusion pump. And you can see how fast it falls, right? This is VLDL turnover. And now the free fatty acids that are coming into the liver are unlabeled, and so the label in the VLDL begins to drop. Here's the study design that is very well known by some of you. This is a 5-day study over almost 20 years ago that we did this study. We recruited patients who were having liver biopsies for diagnosis of non-alcoholic fatty liver disease. And we put them in the hospital for 4 days before their procedure. We fed them an oral isotope. We infused the acetate to label lipogenesis, and we infused the free palmitate to look at the free fatty acids contribution to liver fat. On the beginning of day 4, you can see we did a postprandial study, which I'll show you. This is a bit of a notorious study because we didn't know how to get the dietary triglyceride to the liver. We didn't even know if it would get to the liver if a person ate it. This triglyceride was baked in chocolate brownies, and these poor guys had to eat a chocolate brownie for every meal for these days. In the end, it turns out that when we took the liver biopsy and we looked for that label, we did find it there. I'm going to show you the postprandial study first here. We did blood draws in the fasting and the fed state, right on every day. All right, so this should look a little bit like that mouse experiment because we did the mouse experiment first. We're looking at VLDL triglyceride palmitate in the fasting state at 6:45 and then at 1500 after they've had lunch. And you see this wonderful reciprocal use of fatty acids by the liver in triglyceride. In the fasting state, free fatty acids, which are in the green, are in a high level, and then they drop as the meal had carbohydrate in it. Insulin turns off the adipose tissue, and those fatty acids then are no longer going to the liver. The reciprocal is, of course, the diet. It's present there in the plasma when the person's eaten, and then there's residual the next day. One of the things you can kind of appreciate here is that we were slowly recycling this label over the 4 days. These top triangles are moving up, right, as the body slowly became labeled. The other thing that was quite striking to me is that the pool of lipogenic fatty acids in the liver was quite large in these patients, and it was turning over extremely slowly. It seems like there is some compartmentalization of different fatty acids in the liver. So here is the constant elevated lipogenesis that we observed in the postprandial state after a breakfast meal in these subjects. And I want to contrast that to our publications showing healthy subjects and the stimulation of lipogenesis that one sees by sugars. So in most of us eating a mixed diet, lipogenesis is very low in the fasting state, and then it's wonderfully stimulated after meals, and then it drops again. We didn't see this at all. And I actually wondered if we had performed the experiment incorrectly. And now we've done this in many, many subjects. So if you put all of those sources together in healthy subjects, this is what you see in the postprandial state. You see that stimulation of lipogenesis, you see dietary fat getting back to the liver, and you also see the suppression of adipose-free fatty acid flux to the liver that remains suppressed for 5 hours after this meal. In the NAFLD subjects, you see a rebound of free fatty acids late as the adipose is insulin resistant. You see kind of a low level of dietary fat accrual, and then lipogenesis is high and just doesn't move. We've used water of late to measure de novo lipogenesis, and I wanted to remind you where those carbons or where those hydrogens get incorporated. This is the pathway of fatty acid synthesis. I also want to remind you that one of the products of de novo lipogenesis is CO2 production. And when animals and humans are actively making fatty acids, you can get a respiratory quotient above 1. And I caution you to not put in your paper, if you're using indirect calorimetry calculations in your animals in the fed state, and you get an RQ over 1, and you report that this is negative fat oxidation. And this is not negative fat oxidation, which you will see in some papers. This is net lipogenesis. So it's very likely that your animal in the fed state is both burning fatty acids and making them by DNL at the same time. And when that happens, your respiratory quotient will go above 1. You can use tritium or deuterium to label de novo lipogenesis. In some tissues, there is more or less incorporation of deuterium or tritium. But as I said, we get roughly the same result. The primary product of the fatty acid synthase complex is palmitate. We do see some stearate that has label in it and some myristate as well. Lastly, newly made palmitate and newly made stearate can be unsaturated, and sometimes people use these markers as these fatty acids as markers of de novo lipogenesis. It should be pointed out that the desaturase is elevated by insulin when de novo lipogenesis is elevated. It is not, it's actually possible that the 16-1 that we're measuring is not made directly from de novo lipogenesis, because one could take palmitate from the diet and desaturate it, and you would get high 16-1. All right, I want to show you probably the most complicated human study we did when we tried to measure all of these pathways at the same time. We were interested in nocturnal metabolism, and so we put these NAFLD patients in the hospital the night before their biopsy. We labeled their evening meal with a 16 heavy palmitate mixed into like pasta sauce or something. The next day, we made them miss breakfast and we fed them lunch with a different label so we could track lunch. We infused 13C palmitate to measure free fatty acid flux and free fatty acid contribution to VLDL. And then we used labeled water for 10 days before their procedure to measure lipogenesis. We also, in this case, infused deuterated leucine, and that can be used for protein synthesis or amino acid turnover. So, oh, and I'm reminding myself here, the animation on this slide is intentionally extremely slow because this was the most excruciating experiment I have ever performed in my life, and I just want you to enjoy it. This is de novo lipogenesis coming down from midnight in obese control subjects studied by Jennifer Lambert at UT Southwestern. You can see when we made them miss breakfast in the morning, you were finally starting to get really good fasting lipogenesis levels, and it occurred probably about 10 o'clock or so. The levels then bounced right up when we fed them a meal that had sugar in it. These are the levels from the patients with NAFLD. They are equally obese and equally insulin resistant. And you can see how it takes a very long time for their lipogenesis to actually turn off. So not only have they got twice as much flux through the pathway, but it is resistant to fasting. So I'm going to summarize and conclude here, and then we can entertain any questions, and hopefully the panel will also chip in. I've shown you that various combinations of isotopes can be used to track sources of lipid in tissues in animals and humans, and we've really worked on being able to do this simultaneously. Different types of fatty acids, saturated versus polyunsaturated versus MUFA, have different fates. So if you're going to compare different sources into a single pool, you need to use the same fatty acid and label it differently to denote the different sources. And I'd like to expand here. So if I wanted to see how much liver fat's coming from the diet versus how much liver fat's coming from de novo lipogenesis, I shouldn't label the diet with oleate and then look at palmitate in lipogenesis. And that's because oleate is highly oxidizable in mammals, and its fates are different. So when we're looking at the various sources contributing to a single liver triglyceride pool, since de novo lipogenesis primarily produces palmitate, that's why we've chosen to label dietary palmitate and also infuse palmitate into the blood to track FFA. Thus, for DNL, we give enough deuterated water to the animal or the person such that a newly made palmitate only increases its weight by 1 or 2 on the mass spectrometer. For free fatty acids, we infuse a 4-labeled palmitate. You could infuse a 16 labeled, 6 labeled, whatever. We just want it heavier, right? And then in the diet, we feed a very heavy palmitate, such that when we take the liver sample from the human, take the triglyceride out and look for our labels, if we see a 31 labeled palmitate, we know, that one came from the diet. This one came from the fatty acids. Hope that's clear. Lastly, We label for a long period of time, sometimes days, if the turnover is slow. We label fatty acids that turn over quickly, like the free fatty acid pool, with an acute infusion of label. But because de novo lipogenesis turns over very slowly in the liver, we've gone to labeling a long period of time before we ran our experiment. So I'm going to stop there. I will unshare. I can see my co-panelists there. There will be a recording of this today, and it's going to be posted on the Mouse Metabolic Phenotyping Center site at Vanderbilt. They'll probably show up tomorrow. We can open it up for chat or the panelists. If you have any questions or any comments you'd like to add. Joanne and Steve, can you unmute? Maybe Ben, we can all go to the same, I can stop hosting.
So, Joe, this is really great, Elizabeth. So, a lot of people interested in their favorite fatty acid.
Yes.
So, you mentioned early on that lipid pools are actually localized in the liver, and they're not all the same. So, when you're tracing things where you're interested in these subpools, are there some tricks of the trade you have to worry about in beginning to look at which pool is actually where my palmitate is going? How do I do that?
That's a great question. So good examples of that are you can isolate triglycerides that are associated with the mitochondria or triglycerides that are associated with the mitochondrial membrane, for instance. We've used LCMS to look at a single fatty acid going into phospholipid versus cholesterol ester, for instance. It would be very nice to be able to feed both like oleate and palmitate at the same time, and then to look at their fates that way. But no, it's some of those experiments, you need to pilot them in a mouse, maybe in a couple of experiments, do your subcellular fractionation or your lipid fractionation, and figure out how much label you should be giving. 3 hours of infusion of a free fatty acid is a little bit low to track lipids in mouse adipose. It's great for liver. It works for mouse muscle, mouse heart. But the adipose tissue is big enough. We found we need to kind of go 4 and 5 hours to be able to capture fatty acids turnover and cycling in mouse adipose tissue. Mm-hmm.
So Elizabeth, it's Steve. Hopefully you can hear.
Travis. Yeah, I can.
Hi, Elizabeth. How are you? So can you comment on why virtually all of these studies look at triglycerides, and typically one never really sees data on, phospholipids or cholesterol esters. And I can appreciate, right, a lot of this is to what's the question someone is after. But right, if you look in the literature, there's very little data on the sort of partitioning of fatty acids, partitioning of diglycerides into one pool or the other. Yeah. Can you comment on how the timing of these experiments would lend themselves to looking at other lipid species? Because the liver can make a lot of things. Maybe that would be
sure can. That's a great question. So I have a review paper in JLR, and I think it's probably 2005 or 2006, in which you can see a figure in there where I tracked fatty acids into VLDL, cholesterol esters, phospholipids, and triglycerides at the same time. And this was, I think, a 16-hour infusion in fasting healthy men. And you can see, just like the pictures I showed, you can see triglyceride labeling go up and have beautiful curves. Cholesterol ester, in about half of the experiments we did, it never became labeled in VLDL. And that may be for two reasons. One, that cholesterol ester is transferred into VLDL once it's in the plasma. So the cholesterol ester that's in the particle is coming from a cold, old source somewhere out in the body. Or it could be that the fatty acid that's used to make cholesterol ester in the ER of the liver is coming from phospholipid, and phospholipid is cold. It could be one of those two. For phospholipids, we did see earlier labeling of phospholipid. It started to come up about 4 or 5 hours, whereas triglyceride can be labeled in 20 minutes. And then when you think about ceramide, we're currently labeling ceramide and looking at that labeling because there's, of course, a lot of interest in ceramide. So I think for me, you also need to think about the fatty acid composition of these different lipid species. Phospholipid's going to be highly polyunsaturated. Cholesterol ester is going to have a lot of 18.2, 18.1, right? And so you shouldn't go looking for a fatty acid in some lipid that really doesn't have a likelihood of going there, right?
Finding it, yeah.
Yeah, just because you can put palmitate, let's say, in a phospholipid, you're not doing your experiment right if you're using that to track phospholipid synthesis, I think. Yeah. Can you tell me about, Steve, about kind of what you've thought about potential benefits and limitations of labeled water since you've worked so hard to develop this technique? Could you talk a little bit about that?
Yeah, so it's a good question or a good topic, right? If people are getting started in this area. So typically what would be the alternative would be carbon labeled acetate or glucose or something. So, you know, in some experiments, those can be given orally. So it's not a huge burden that one has to put a catheter into if you're doing an animal study or humans for that matter. But water is definitely, on the practical side, it's easier to administer it, right? So in that sense, it's a lower bar to getting started. The trade-off is that if we've done this experiment in animals before and in humans too, is that deuterium can have some isotope effects. These can be transient. They're not going to last forever necessarily, but right, the D2O does taste different than regular water. And so if you're doing this, we've done non-human primate studies, this can be a little tricky sometimes to get them to drink on their own. And even in humans, right? They may not like the taste of it. So one can mask the taste of this by putting artificial sweeteners if you want, or something in there if that's compatible with the study, and you can do that. So I would say the thing you look at the beginning is how are you going to get the tracer into whatever the subject is, you know, you're working with. And water is definitely easier to get into a subject. And water is nice because it goes everywhere. The caveat though is that it's hard to give a lot of water, so it does put pressures on the measurements that you try to make later on, and that can become a problem. But as you said, there are ways around this. There are different analytical instruments people can use. Depending on the time course you're running your experiment, maybe putting a low dose of water is not such a problem because you run the experiment for a long time and you can see label accumulate. So I would say, go ahead.
One of the problems for a mixed molecule like ceramide, we found that water is wonderful because it goes everywhere, and water is terrible because it goes
everywhere.
Right. And so in triglyceride, it could label the backbone of glycerol and the fatty acids, right? And so sometimes you get overlapping molecular weights, which are difficult to discern. And so I think we have to be pretty careful. If you're going to start an experiment using labeled water, that's the only thing you should use. I would not advocate anybody adding a labeled fatty acid or another label. Just do your first experiment clean and figure out, you know, kind of the labeling pattern that's going to show up from labeled water. It might be way more complicated than you anticipated. We've seen that a number of times.
Yeah, so it is an interesting point, Elizabeth, about where a tracer lands in a product, right? So when you, like, I think probably most of the stuff you showed today was you isolate the triglyceride, then you degrade it, and then you put the fatty acid back into the mass spectrometer. So you physically separate, you know, the backbone from the fatty acid before you do the analysis. So that's nice because that can help with separating out, is my label in this part of the molecule or that part of the molecule. That can definitely help. It does become interesting now with advances in mass spec technology or NMR for that matter too, that you can locate sometimes where the isotope is, if it's in a backbone of something or if it's in a, you know, fatty acid. But I do agree with you that if you're playing with deuterium, the deuterium can sometimes move when molecules fragment and so forth. So you do have to be a little bit careful with that. But I think the people on the call, I'm sure, advancing technologies and stuff, a lot of people are thinking about this now. So some of these problems I'm sure can be solved. If not solved already, will be addressed soon enough.
Sure, sure. We've got a question in the chat, and if anybody else wants to add a question, they should feel free to go ahead. One of the experiments someone's asking is Dr. Sofer is asking why I fasted subjects from 6 p.m. to 12 noon the next day to measure VLDL, any effect of fasting on the outcomes. And yes, that was my whole goal. What we find is that people that are insulin resistant fast pretty well, actually. It's eating that uncovers insulin resistance, and nocturnal post-dinner metabolism is quite aberrant in patients that are insulin resistant. So that experiment was designed to test how subjects were processing the evening meal throughout the night, and then the next day, how did they respond to fasting? And in this experiment, we actually repeated it from baseline to 6 months after they'd lost 10% of their body weight. And one of the things we observed with this missing breakfast kind of long fast is that before they lost weight, their adipose tissue was quite reactionary. And right at 8 o'clock, the adipose just started pouring fatty acids out into the blood. And they were missing breakfast. They were probably hungry and a little bit anxious. After 6 months of weight loss, and then we worked really, really hard to get 10% weight loss, when we fasted them, their adipose tissue was quite well behaved. It continued to release the fatty acids they need. It didn't over-release fatty acids. And then when they ate, we had excellent suppression of fatty acid release after the meal. So going for the long fast was intentional to measure fasting metabolism because you can really see a change before and after weight loss. Let's see. Here's a question. Let's see. I think that's it for questions at this point.
So Elizabeth, this is Owen.
Yeah.
So one of the challenges working with really small animals like mice is that lipolysis and picking up the animal work very well together to stimulate lipolysis.
Yes.
All right. So one of the issues we've always thought about is, so you do an experiment, you would take away food and make them skip their breakfast, which is at night, and come back tomorrow morning, and you actually get a fasting steatosis.
Yes.
And could you explain what fasting steatosis is? And is that steatosis the same as what we would call steatosis related to obesity or metabolic disease?
Yeah, it really is not. Scientists need to think about their animal, and the mouse doesn't eat meals. The mouse eats constantly, right? Eat, rest. They're over to that food cup all the time. That's because the amount of food they can consume in a certain period of time compared to their metabolism is small. So they must eat many, many meals during the night. And so their system is not set up to fast. Mice don't fast well at all. When you start to take away their food, adrenergic stimulation causes those fatty acids to just pour out of adipose tissue. So they are highly stressed with fasting. Whereas in a healthy human, if you miss breakfast, your free fatty acid concentration doesn't change one bit. If you miss lunch, it starts to rise a little bit. But we don't even as humans begin to make ketones until the end of a day of fasting. So humans, because we eat meals, we're fasting fed, we're very good at fasting. Whereas mice, being a carbohydrate consumer, living mostly on carbohydrate, is not set up. Their system is not set up. So that will cause a steatosis, stressing an animal, fasting an animal in some mouse models, but not all, which I find quite interesting. And then the other issue about that is a free fatty acid fatty liver. Whereas insulin resistance, when you overfeed animals with high fat diets and sucrose, lipogenesis is highly stimulated, and that's going to increase lipid stores in liver. So very, very different. I'm really an advocate for acclimating the animals and trying to reduce the stress-induced effects on your gene expression as much as possible. All right, I'm going to read another question here. Is there evidence of futile cycling of labeled fatty acids going through DNL and turning back into palmitate? After all, both lipids and glucose contribute to excess citrate, acetyl-CoA for DNL in the liver. So I'm going to interpret that to mean, do we make fatty acids and can we turn around and burn them to make acetyl-CoA again? And the answer to that is yes. And there have been a really nice papers about 2 years ago showing that this is the case. So on the whole, when an animal is making fats, they're not burning them very much. And when they're burning fatty acids from adipose, they're not making new fatty acids from glucose. But on the whole, all you're doing is turning up a rheostat. So nothing is ever completely off. It's quite surprising to us that insulin resistant humans don't turn off de novo lipogenesis very well at all. It's kind of constantly on, and it appears like there's a ceiling. We have seen some patients with fatty liver who 60% of the fat in their liver is coming from de novo lipogenesis, but that's kind of high. Typically, it's 30% or so. There is a question from Jean-Marc Schwartz saying, are DNL measured by labeled acetate and D2O comparable? We have done repeat studies in subjects, and it depends on, as you know, Jean-Marc, how long you're labeling. If you label with acetate long enough, and you could feed or infuse, as he has shown, and you get turnover, if you only do it a short period of time, your apparent DNL is going to be low. If you label with water for a very long period of time, then you're going to get a higher apparent DNL. I'm going to ask Joanne to start to think about kind of mistakes people make when they start in this field. You don't have to come on or answer now, but I think that would help me a lot. I think a lot of the faculty in the isotope course assist people trying to plan their experiments, and we try to keep people from making kind of the easy experiments. Have you got any comments in that regard?
Well, you know, the biggest mistakes you can make are the physiological ones that you've gone over some of those. And Owen is an expert at the not understanding exactly what your animal is doing, or assuming you can do things that may seem reasonable, particularly if you're thinking of humans and then moving to mice or vice versa. So I think the biggest issues are always the physiological ones, but there are lots of little tracer issues that one may not be aware of, or may not do as good a job as they could dealing with. And those are calculation wise, or planning a tracer study, I think is something that need to put a lot of effort in that. Obviously, people can see here how much work these studies are that you've done. These are elegant studies, and they are a huge amount of work. And if you miscalculate or don't do enough preliminary studies to get enough isotope to where you need to get it to see it, as Steve was saying, if you don't have your water high enough, then your calculation will have a lot of error in it, and you know how valuable it is. So I'm of course I'm a big advocate of doing a simulation of it if you can. Try out some calculations. If we get this and if we get that, try some pilot studies. Make sure the isotope, you've got enough isotope to see, because you can't see it. Really, that's the worst thing. But there are lots of things, and I think coupling together the planning for the good physiology and good isotope studies is going to be very important. And perhaps work together more on this as in the course.
The very first experiment, as I mentioned, that we did, nobody had ever tracked dietary fat into the liver in a human. And I went to the rodent literature. Well, rodents, they don't put much of their dietary fat in their liver, and they don't have much dietary fat in the first place. So they drop off about 95% of the dietary fat to the periphery. And that tiny little chylomicron with a teeny bit of dietary fat left in it goes back to the liver. So we loaded up those brownies with so much label. I am telling you, I was so afraid we were going to get the precious liver biopsy and never see a dietary. Well, humans eat fat all the time. And as we know from even movies, you can get fatty liver by eating a lot of dietary fat. And when that tissue came into the lab and we put it on the mass spec, I just about fell off my chair because the dietary fat label was so huge. And so to me, I think that's the way you do it. You go to the literature, you estimate what you think it's going to be, and then you perform your pilot experiments to get a sense. Those were very expensive experiments, and right away I figured out, we don't have to put so much label in those brownies. We can reduce the amount. Someone is asking, what are the carbon sources for increased lipogenesis in insulin-resistant patients? Well, there have been a number of studies that have reduced sugars in people's diets, and liver fat will drop very, very nicely. Whether or not glucose is as lipogenic as fructose, I think that there are very good data to suggest it is not, that fructose is highly lipogenic. And so we believe that it's simple sugars in the diet that are primarily driving fatty liver in insulin-resistant patients. And this brings me to the very good question by Jamie Young from Vanderbilt, who's on here, who says, in a disease like NAFLD, a naive question might be, isn't all fat just fat? Why do you care about where it comes from? From a clinical standpoint, why do you want to know where the liver fat is coming from? Well, there are no treatments for fatty liver disease. And if we can figure out the sources, we can figure out where to target the treatment. If adipose insulin resistance and free fatty acid flux is causing fatty liver, then a drug like pioglitazone that might reduce fatty acid flux could reduce liver fat. And that has been shown to be the case. For if we need to treat with a diet, should I reduce dietary fat or should I reduce dietary carbohydrate? And we have found that it's much more efficacious to reduce dietary carbohydrate. Thus, lipogenic fatty acids are different than plasma-free fatty acids. A palmitate is not a palmitate. And that is because when de novo lipogenesis is on, this sends messages, global messages throughout the liver to re-esterify any fatty acid that comes to the liver. And I'll give you an example of this. There's a very nice paper that shows when fat is consumed, I think it's by Chung, when fat is consumed alone, dietary fatty acids are burned. When those same fatty acids are consumed with a carbohydrate and lipogenesis gets turned on, the same dietary fatty acid gets stored, not burned. So lipogenesis being on tends to reroute fatty acids from any other source to the triglyceride droplet and causes an increase in storage. All right, if Steve is still on, I can also answer this, but the question is, when using deuterated water, when liver DNL can reach the steady state, do you think 1 week is long enough? Well, it depends on what you're trying to measure and how much liver fat is there. We had a patient who we calculated her liver fat turnover was 2 months. She had so much fat in the liver and the turnover was so slow. And so you want to label, we used 10 days. A recent paper by Sam Klein's group used much longer than that because they were also trying to label protein or fibrosis synthesis as well.
Right.
So Steve, do you have any
Yeah.
comments?
No, Elizabeth, it's a very good question. And I think, yeah, it seems like a week or 2 weeks maybe is what people are talking about now, right? Yep. Mark Hellerstein has some papers on this as well. It does seem kind of strange if you think about like, well, why should it take that long to get stuff in and out of the liver when there's so much traffic flowing through there? But it is what it is, I guess. And so it can take a while. I would add one comment though too, Elizabeth, that I'm not sure one has to always wait to find, the true number. Is it 40% DNL or is it 30% DNL? It depends on what someone's question might be. If you're playing with, potential drug development, maybe you can get away with, 5 days and you can see yes or no, my compound of interest or my target I'm trying to manipulate is doing something. You may not get the right absolute answer, but at least you can pick up relative trends. So I'm not sure you always have to wait that long.
I agree.
The one nice thing I would add though, the one nice thing with the D2O is that it's not that difficult to run a long experiment because it's so easy to administer the tracer. Yeah. And so on the flip side is that so much time and effort is going to go into setting up the experiment. If you're debating, do I need 1 week or 2 weeks? It may sound like adding 2 weeks is twice as long, but in reality, it's not that much more work to run it a little bit longer. But again, it's going to depend a little bit on the specific context of the question, I would say.
Yeah, I totally agree. And you can just put it in the water bottle of the mouse, right? And if you want to look at strain differences, you may not get complete turnover, but if you have a knockout mouse, you can, as long as you treat those animals the same, then you'll have a comparator, that's for sure.
Elizabeth, this was a point I want kind of linked to what you were saying, this idea of the percent of a pathway that's DNL versus the absolute rate of DNL. And a lot of people, percent is easier to measure.
Yeah.
Absolute rates are hard to measure.
Yes. Yeah.
Could you speak to kind of like this idea of what are the issues one has to think about when you think about it going from contribution versus absolute flux?
Right. And maybe you can cover this again when you're thinking about gluconeogenesis and hepatic glucose production next month.
Yes.
Because it's the same. That's right. So why do I care about the fractional de novo lipogenesis? Well, we find that the percent of palmitate made from this pathway is predictive of liver fat over and over in little kids across ethnicities, doesn't matter what age, right? So the fractional DNL is quite predictive in terms of predicting insulin resistance and the quantity of liver fat. The absolute quantity is lower. And this is the thing that people get confused and ask me all the time. The primary source of fatty acids coming out of the liver is from the plasma-free fatty acid pool. That's the primary source. But to my knowledge, the flux of that pathway doesn't control the other sources of fatty acids, whereas the flux through DNL changes the way the liver handles free fatty acids and dietary fatty acids. So that's why. So de novo lipogenesis is a lower quantity of fats in the liver and in VLDL, but it is a primary controlling point in metabolism, and that's why we care.
And a way to flip that around would be, even if it doesn't control a process, it reflects a controller of a process that has changed.
That's what I mean. And
we don't know which.
Yeah,
right?
we think of malonyl-CoA. When you're making fatty acids, you're making malonyl-CoA. Malonyl-CoA can turn off global fatty acid oxidation in the liver. So it is a pathway that, when it's on, tells the liver, we're fed, we're fed, we're fed, we're fed. Store everything that comes to you. And that pathway is on in the middle of the night in people with fatty liver, at 8 in the morning, it's constantly on. And so pointing towards storage is really the problem here. And when we cut sugars out of the diet, then that pathway is turned down and liver fat immediately begins to fall. I'll end here with one last question. Somebody wanted to know, if you reduce dietary sugars and liver fat falls, does weight always fall? And weight will come off people when they cut sugars out of their diet, that's for sure. But John Mark Schwartz has a beautiful paper showing that at weight stability, even if you reduce sugars, you can lower liver fat. So that really points to this controlling nature of that metabolic pathway for global metabolism in the liver.
Yes.
So we should stop here. Thank you for all in the audience for being with us today, and watch for Owen's talk in about 2 weeks' time. You can find all this information on the Vanderbilt Mouse Metabolism Phenotyping website. And look for us on YouTube tomorrow. Bye.
Bye.